Hi everyone. Today's lecture is on part 2 of amino acid metabolism and urea cycle. The key reactions of amino acid metabolism are transamination and oxidative deamination. Transamination reactions. Generally, it is the first step of amino acid degradation. Transfer of the alpha amino group from an amino acid to an alpha keto acid, resulting formation of a new amino acid and a new keto acid. Transamination reactions are reversible and catalyzed by aminotransferase enzyme, which requires pyridoxal phosphate as cofactor. Pyridoxal phosphate derived from vitamin B6. Example for a transamination reaction. Alanine combined with alpha ketoglutarate in the presence of alanine aminotransferase converted to pyruvate and glutamate. Again, pyruvate and glutamate can combine together, can form alanine and alpha ketoglutarate. This is catalyzed by alanine aminotransferase enzyme. Aminotransferases. Aminotransferases are found in both cytosol and mitochondria. They are used for both synthesis and breakdown of amino acids. Transamination reactions tend to channel amino groups onto glutamate. Two most important aminotransferases. Alanine aminotransferase or ALT. Alanine is important in muscle where 25% of amino acid nitrogen is transported out on alanine. In liver, reverse reaction moves amino acid nitrogen back on glutamate. Aspartate aminotransferase or AST. Aspartate important in liver since half of urea nitrogen is from aspartate. Some of the example for transamination reaction. Alanine combined with alpha ketoglutarate in the presence of alanine aminotransferase converted to pyruvate and glutamate. Likewise, aspartate combined with alpha ketoglutarate in the presence of aspartate aminotransferase converted to oxaloacetate and glutamate. Transamination reaction can occur in two steps. For an example, if an amino acid combined with alpha ketoglutarate converted to keto acid and glutamate. The glutamate is the new amino acid formed by this reaction. Glutamate again combined with another alpha keto acid such as oxaloacetate in the presence of aspartate aminotransferase can be converted to alpha ketoglutarate and aspartate. Aspartate enters urea cycle. Aminotransferases clinical correlation. Two specific transaminases are of clinical importance. Alanine aminotransferase or ALT is a cytosolic enzyme. Aspartate aminotransferase or AST occurs both in cytosol and mitochondria. Increased amounts of both transaminases leak from inflamed or damaged hepatocytes. Alanine aminotransferase is more specific for liver disease than aspartate aminotransferase, while latter is more sensitive because the liver contains larger amounts. Aspartate aminotransferase is elevated in acute myocardial infarction. Disposal of amino group deamination reactions. Glutamate dehydrogenase. Glutamate in the presence of glutamate dehydrogenase, undergoes an oxidative deamination reaction to produce alpha-ketoglutarate. In this reaction, glutamate is oxidized and an amino group is removed from glutamate. Thus, it is known as oxidative deamination. This reaction also reversible. Glutamate dehydrogenase is one of the enzyme utilizes NAD+ and NADP+ as cofactor. ATP and GTP allosterically inhibit this enzyme. ADP and GDP allosterically activate the enzyme. When the energy levels are low, amino acids are deaminated to provide alpha-ketoglutarate for the TCA cycle to generate energy. Big picture. Alpha amino acids in the blood or in the body derive from diet and body protein. These alpha amino acids, in the presence of aminotransferases, combine with alpha-ketoglutarate forms alpha-keto acids and glutamate. So all the amino group is transferred to glutamate now. Glutamate undergoes oxidative deamination reaction which is catalyzed by glutamate dehydrogenase. And during oxidative deamination, alpha-ketoglutarate and ammonia are formed. Glutamine, by the action of glutaminase enzyme, can be converted to glutamate and ammonia. Ammonia in turn can combine with glutamate in the presence of glutamine synthetase converted to glutamine. The degradation of purine and pyrimidine and other various nitrogen compounds also result in ammonia formation in the blood. Urea present in the intestine by the action of bacterial urease also converted to ammonia. Ammonia in high amounts is toxic to the brain. So ammonia has to be converted to a less toxic compound. Urea is less toxic than ammonia. So in subsequent reactions, liver converts ammonia to urea. These reactions are known as urea cycle. Urea cycle was the first metabolic process to be described as a cycle by Sir Hans Krebs, who also described the TCA cycle. Role of urea cycle: rid the body of toxic ammonia, therefore permitting the use of amino acid as an energy source. Liver, major site of urea synthesis and major source of arginase, and is the only tissue with a complete set of all 5 enzymes required for urea synthesis. Other tissues have enzyme for reaction 3 and 4 only to make arginine or nitric oxide, which are important in blood pressure, neurotransmission, and macrophage antibacterial action. Compartmentalization of the urea cycle. Urea cycle takes place in the liver periportal cells. It takes place in 2 intracellular locations and consists of 5 reactions. In the mitochondrial matrix, carbamyl phosphate formation and citrulline synthesis take place. In the cytosol, arginosuccinate formation, cleavage of arginosuccinate to arginine and fumarate, hydrolysis of arginine to ornithine and urea take place. Urea contains 2 nitrogen atoms. One nitrogen is supplied by ammonia formed by the transdeamination of amino acid. The other derived from aspartate. The cycle uses a carrier molecule which is regenerated. Ammonia detoxification by the liver. Periportal hepatocytes contain urea cycle enzyme and glutaminase, whereas glutamine synthetase is located in perivenous scavenger cells. Urea and glutamine synthesis are anatomically switched behind each other and represent functionally the sequence of a periportal high capacity but low affinity system and the perivenous high affinity system for ammonia detoxification. Periportal glutaminase acts as pH modulated amplifier of the mitochondrial ammonia concentration and is an important determinant of urea cycle flux. Ammonia that escape periportal urea synthesis, the low affinity system, is disposed of by the perivenous scavenger glutamine synthetase, the high affinity system. This guarantees the effective detoxification of ammonia even urea cycle flux is inhibited in acidosis. Reactions of urea cycle. The production of urea from ammonia involves 5 enzymatic steps. The urea cycle begins inside the mitochondria of the hepatocytes, but 3 of the steps occur in the cytosol. The cycle thus spans 2 cellular compartments. The first amino group enter the urea cycle is derived from ammonia inside the mitochondria, arising by the multiple pathways described in the figure. Some ammonia also arrives at the liver via the portal vein from the intestine, where it is produced by bacterial oxidation of amino acid. Whatever its source, the ammonium ion generated in the liver mitochondria is immediately used together with bicarbonate produced by mitochondrial respiration to form carbamyl phosphate in the matrix. This ATP-dependent reaction is catalyzed by the enzyme carbamyl phosphate synthetase 1. The mitochondrial form of the enzyme is distinct from the cytosolic form, which has a separate function in pyrimidine biosynthesis. Carbamyl phosphate synthetase is a regulatory enzyme of urea cycle. It requires N-acetyl glutamate as a positive modulator. Carbamyl phosphate may be regarded as an activated carbamyl group donor. The carbamyl phosphate now enters the urea cycle, which entails 4 enzymatic steps. Carbamyl phosphate donates its carbamyl group to ornithine to form citrulline and release inorganic phosphate in a reaction catalyzed by ornithine transcarbamylase. The citrulline is released from the mitochondrion into the cytosol. The second amino group is introduced from aspartate, which is generated in the mitochondria by transamination and transported to the cytosol. Aspartate condensation reaction between the amino group of aspartate and the urido group of citrulline to form arginosuccinate. This reaction catalyzed by arginosuccinate synthetase of the cytosol. It requires ATP. The arginosuccinate is then reversibly cleaved by arginosuccinate lyase to form free arginine and fumarate. Fumarate enters the pool of citric acid cycle intermediates. In the last reaction of the urea cycle, the cytosolic enzyme arginase cleaves arginine to yield urea and ornithine. Ornithine is thus regarded and can be transported into the mitochondrion to initiate another round of urea cycle. Regulation of urea cycle. Urea cycle regulated by short-term regulation and long-term regulation. Short-term regulations are substrate availability and allosteric regulation. Long-term regulation is induction of urea cycle enzyme. Substrate availability. The higher the rate of ammonia production, the higher is the rate of urea production. This is feed-forward regulation of urea cycle. Factors known to increase urea production: glucagon, ammonia, increased amino acid load, alkalosis, activation of glutaminase, mitochondrial bicarbonate supply. The activity of the urea cycle is allosterically regulated at two levels. The key regulatory enzyme of urea cycle is carbamyl phosphate synthetase 1. It catalyzes the reaction between bicarbonate and ammonium ion to produce carbamyl phosphate. This is an energy-consuming reaction. Carbamyl phosphate synthetase 1 allosterically activated by N-acetyl glutamate. N-acetyl glutamate, in turn, produced from acetyl-CoA and glutamate. The reaction is catalyzed by N-acetyl glutamate synthase. The reaction catalyzed by N-acetyl glutamate synthase is allosterically activated by arginine. Thus, the activity of the urea cycle is regulated at two levels. Long-term regulation of urea cycle. 5-urea cycle enzyme and N-acetyl glutamate synthase levels decreases with low protein diets and increases with high protein diets. During starvation, increased amino acid catabolism. Therefore, the level of this enzyme increases and increased urea synthesis. Increased enzyme synthesis take place at gene transcription level. Decreased enzyme degradation. The changes takes place over 3 to 7 days. 20 to 30-fold increase in enzyme concentration during starvation. Ammonia toxicity. Ammonia, one of the most toxic compounds produced by the body. Elevated levels of ammonia is known as hyperammonemia. The symptoms of ammonia intoxication: tremors, slurred speech, blurred vision. At very high concentrations, ammonia causes irreversible brain damage, coma, and death. Proposed mechanism of ammonia toxicity. Ammonia readily traverses the blood-brain barrier. In the brain, is converted to glutamate via glutamate dehydrogenase, depleting the brain alpha-ketoglutarate. As alpha-ketoglutarate is depleted, oxaloacetic acid falls correspondingly and TCA cycle activity comes to a halt. ATP production via oxidative phosphorylation stops. Irreparable cell damage and neural cell death occurs. Increased glutamate leads to glutamine formation. This depletes glutamate stores, which are needed in neural tissues. Glutamate is a neurotransmitter and precursor for the synthesis of gamma aminobutyric or GABA, another neurotransmitter. Reduction in brain glutamate affect energy production and neurotransmission. Proposed mechanism of ammonia toxicity. Elevation in neural glutamine concentration. Glial cells volume is controlled by intracellular organic osmolyte metabolism. Glutamine serve as organic osmolyte. As glutamine level rise in the brain, the volume within the glial cells increases, resulting in formation of cerebral edema. Disorders of amino acid metabolism. Phenylketonuria, albinism, alkaptonuria, maple syrup urine diseases, histidinemia. Very rare inborn errors of metabolism result in severe development abnormalities if left untreated. Enzyme deficiency in alkaptonuria, albinism and phenylketonuria shown in the figure. Phenylalanine hydroxylase deficiency results in phenylketonuria. The tyrosinase deficiency results in albinism, and homogentisic acid oxidase deficiency results in alkaptonuria. Molecules derived from amino acids. Nucleotide, nucleotide coenzyme, porphyrins, neurotransmitters, and other molecules. Synthesis of epinephrine, norepinephrine, GABA, histamine, and serotonin. Epinephrine and norepinephrine is synthesized from the amino acid tyrosine. Tyrosine is converted to DOPA by tyrosine hydroxylase. DOPA converted to dopamine. Dopamine is converted to norepinephrine. Subsequently, norepinephrine is converted to epinephrine. Glutamate, by decarboxylation reaction, catalyzed by glutamate decarboxylase, is converted to neurotransmitter gamma-amino butyrate. Histamine is derived from amino acid histidine by decarboxylation catalyzed by histidine decarboxylase. Serotonin is derived from tryptophan. Tryptophan initially converted to 5-hydroxytryptophan, subsequently by decarboxylation by an aromatic amino acid decarboxylase converted to serotonin. Synthesis of creatine and glutathione. Creatine is synthesized from two amino acids, glycine and arginine. Glycine and arginine combine together form guanidinoacetate. Guanidinoacetate in the subsequent reaction is converted to creatine. Creatine phosphorylated by ATP to form phosphocreatine. Glutathione is synthesized from glutamate. Glutamate combined with cysteine in the presence of gamma-glutamyl cysteine synthetase forms gamma-glutamyl cysteine. Gamma-glutamyl cysteine in the presence of glutathione synthetase combined with glycine forms reduced glutathione.